Oh, yeah. I could move in the back there. You want me to? Huh?
Yes. Oh, you want to? Oh, you can go around.
No?
Yes.
Yes.
Yeah.
Okay.
What you are looking at here are several power supplies for the instrument. This first one provides the high voltage for the photomultiplier. Photomultipliers need voltages around 1500 volts or more. This panel underneath supplies the energy for the mercury arc lamp. And then the next one here is the power amplifier for the oscillator unit, which is at the bottom. This oscillator unit drives the separator that is over here. So
Now, you mentioned the mercury arc lamp. Isn't it true that very soon after the first instruments were developed, that they started using lasers rather than
yes,
arc lamps?
this mercury arc lamp, as a matter of fact, had very little use. I used it for about a year or two and then switched to a laser. The reason is that mercury arc lamps flicker all the time, and they are very bad for the instrument because the moment it flickers, then one loses the focus, and they were a real pain. And the lamps burnt. Usually, in the best experiment is when they burn. So we kept on using fresh lamps that were very expensive, and we were not happy at all with that. The laser simplified the instrument considerably. But I have put here the original lamp, so the instrument is authentic.
Hold on a second. Explain it just briefly. This is the lamp.
This is a mercury arc lamp that illuminates the cells flowing through the chamber. The mercury arc lamp was replaced later on with a laser. The reason was that these mercury arc lamps are very unstable. They flicker and they get bad usually in the best part of an experiment. So, but I have not put here the laser because I wanted to keep the authentic instrument at this time.
We're at a much better angle for that.
Okay.
Boris?
Yeah. We know when.
I'll say.
This is the most important part of the instrument. It's a flow chamber and has 4 ports on each side. One is here. The other one is facing the microscope, where one observes what's going on in the flow chamber. The third one looks at the mercury arc lamp, and the fourth one at the photomultiplier. These are the inlets for the water to clean the windows, and the cells flow from underneath, and the waste comes here from the top. The photomultiplier has a filter to exclude the exciting blue light that comes in from the mercury arc lamp. The mercury arc lamp has its own filter, which makes the light monochromatic. These are adjustments for the lenses. Again, too many adjustments was difficult to get a good focus.
Just those again, just so you can see.
And also one could adjust this lens that focus on the photomultiplier. Then at the end of the photomultiplier is a preamp that is to increase the signal voltage, and then it goes to a larger preamp, which is this box here, and from this preamp goes into the analyzer where it is then cleaned up and shown in the oscilloscope.
Good.
The pulses from the photomultiplier go through two preamps, a small one at the end of the photomultiplier and the one that is on the left of the analyzer. From the preamp, the pulses go directly into the analyzer where they are sorted and shown in the oscilloscope.
Good. I think we got everything.
We will now be speaking with Dr. Mack Fulwyler, the inventor of the cell sorter. Mack, why don't we start with the impulse for even thinking about a machine to sort cells?
It was the result of not a clean scientific inquiry into an area of knowledge or the addressing of a needed tool. It really arose from less admirable motivation. When I first went to Los Alamos and went to work under Marvin Van Dilla, our primary interest was in monitoring the fallout from atmospheric nuclear weapons testing. That fallout appearing in milk and meats and other food products. With the arrival of the Atmospheric Nuclear Test Ban Treaty, the appearance of fallout, radioactive fallout, in the diet and in humans diminished. And so we had much less to do in terms of monitoring fallout. So our group, which consisted of 4 individuals, 3 physicists and a physical chemist, looked around to see what we could do to assist the biologists who were the other 60-some people of the group. Marv and I then became interested in exploring, at first, a Coulter counter, which is a device that's commonly used even now in hospitals to analyze blood samples, getting a blood cell count and measuring the size of blood cells so that you can determine if an anemia is present. A Coulter counter uses a small orifice in a glass tube. The cells are suspended on one side of that orifice and then sucked through the orifice by a vacuum. An electric current is also passed through that small orifice at the same time the cells are passing through. And the voltage across the orifice is measured. As a cell passes through the orifice, then, it obstructs the flow of electric current, and you measure the electrical signal that results from that. The presence of the signal says that there's a cell going through. The amplitude of that electric signal tells you the size, the volume of the cell going through. So The device allows you to run a sample of cells through it and to obtain a distribution histogram that represents the size of the cells in that population, the range of sizes of the cells in that population. We had a pathologist in our group who was using that device to analyze blood, and he would adjust the aperture current and some other characteristics of the machine in such a way that he could cause a small subpopulation of the red blood cell distribution to move away from the main distribution of red blood cells. And the pathologist thought that that represented immature red cells that had just been produced. Well, Marv and I did not believe that that was the case, and we set out to try to convince him that he was incorrectly using the device, and we were not successful in doing that. It occurred to me that if I could just pick out this, what was thought to be abnormal population, just physically isolate those cells, run them back through the Coulter counter, get the same distribution, it would demonstrate that it was an artifact that he was misusing the machine. So my motivation in trying to come up with a way to sort cells was to disprove this fellow's interpretation of data. So at that stage, I set out to look for ways to physically isolate cells based on some electrically generated signal. And I started off looking at valves, mechanical valves, which could be electrically switched and move a flowing stream of liquid from one channel into another channel and then back. But when you look into that, it's a very slow process, and you could not process cells very rapidly. Then I saw a paper by Dick Sweet, who at that time was at Stanford Research Institute, working on I believe it was a Navy project, maybe an Air Force project, to develop an ink writing oscillograph, which is a device that produces a jet of ink in the air. The jet is vibrated so that the ink jet breaks down into droplets. The droplets can be charged according to an electronic signal that's coming from something you want to analyze. The charged droplets then entered in a deflection system where they were deflected, and then they impacted on a moving strip of paper that passed underneath the system. So these moving streams would write out the signature of the electrical signal coming through the device. Well, I saw that as a way to move small amounts of liquid very rapidly. And so what I put together initially was the idea of using a Coulter volume sensor, which generates an electronic signal proportional to the size of the object, and this ink writing oscillograph, which partitioned a stream of liquid into small droplets, which could then be charged and individually deflected. So I saw these small droplets as really just carriers for a cell, as a means of moving a cell around. So that was the initial idea. I put out a letter describing the process to a number of scientists in the laboratory, Los Alamos Scientific Laboratory. And I've forgotten the exact number, but I think about 8 people I asked for their opinions on whether or not the method would work. The majority came back saying, no, it probably wouldn't work for various reasons. But under the encouragement of my boss, Wright Langham and Marvin Van Dilla, I went ahead and tried it and made a trip out to visit Dick Sweet at Stanford Research Institute, and he was very cooperative, very supportive, thought the idea would work, and loaned me some of his devices for making ink jets. And his devices for making ink jets consisted of a hypodermic tube with a small glass nozzle inserted in the end of it so that you could generate a stream of ink of about 60 microns in diameter. Then he vibrated the metal tube with an electrostatic oscillator, and the vibrating needle would then produce droplets. I borrowed that system from Dick and tried to build a Coulter counter into the hypodermic needle and had all kinds of troubles, could never get that to work. So then I tried other ways of getting a Coulter volume sensing orifice and the two electrodes that you need, one on each side of the orifice, into a fluidic system where I could also apply sonic energy into the liquid so that I could break the stream into droplets. And this device is probably the second or third version of my efforts to make a flow cell which would both measure Coulter volume and produce droplets. One of the problems in measuring Coulter volume is that you're trying to pulse a system vibrated at a frequency of perhaps 60,000 vibrations per second. At the same time, you're trying to measure very tiny signals that are being generated in the midst of this moving system. And by simply moving the liquid passing through the device, there are changes in capacitance and other changes that produce electrical signals. So one of the big problems was to isolate from the electrical measurements the effect of the acoustic energy vibrations in the system. And this is one of the ones that worked. There's a piezoelectric crystal mounted in this part of the system. It's 1 inch in diameter and ½ inch thick. And it's driven by the sine wave generator and the amplifier that Dr. Rotman described earlier. The energy, the vibrational energy produced by the crystal is coupled into an aluminum rod here, which rests right up against the crystal. And from the aluminum rod into this plastic horn, this is called an acoustic horn. It's a catenoidal shape. And the reason for that is that up at the top, if you look at the range of motion, the number of microns or fractions of a micron of physical motion resulting from the piezoelectric crystal, it's very small. And so this shape of an acoustic horn is like a transformer in that it increases the amplitude of the motion, even though a smaller amount of mass is moving. It increases the amplitude of the motion. Within this portion, the end of the plastic rod is in direct contact with the liquid. And so you're coupling the acoustic energy right into the liquid cell suspension, which then jets out of a small orifice in a platinum disc down here. The electrodes for the system, and using the Coulter counter principle, inside of this is a large piece of platinum, which is in contact with the solution. Then there's a small glass orifice, which is the volume sensing orifice. Then there is a platinum disc in the end, which establishes contact on the downstream side of the liquid jet. Coming out of the platinum disc then, through a small drilled orifice of about 60 microns, you have a liquid jet which travels down for perhaps a centimeter or so, and then, because of the action of the piezoelectric vibration, it breaks into droplets. This is all shielded, so that all of this metal here serves the purpose of shielding the sensitive electrical electrode from influences, electromagnetic influences in the surrounding. So this was one of the first ones that was able to work fairly well. So that pretty much describes this system.
What don't we see here? We don't see the difference.
Okay, Dr. Rotman's purpose with this system was to simply use it as a generator of uniform droplets. To have a true cell sorter, this is missing two deflection plates, which are the high voltage plates, and which cause actual deflection of the charge droplets, and then some kind of a collection system down here.
Okay. All set.
Now that you've described the sorting mechanism, could you tell us, Mack, what you actually patented?
Okay, the patent, which issued in, I think, 65 or so, the patent covered the concept of analyzing a cell, a flowing stream of cells, analyzing the cell by any electronically convertible means. And it specifically mentioned Coulter volume, fluorescence, radioactivity, and a couple of others that I don't even remember. Means of sensing cellular characteristics, light scattering was one of them. But in principle, any means of electronically extracting a measurement from a cell could be used to trigger this system of sorting. One of the advantages is that the sensing element is completely divorced from the sorting element. And there is a time delay in between the two, so that you can bring out a measurement, run it through an analysis, and compare it with standards for sorting or not sorting, and then make an electronic decision whether or not to cause the sorting event. So it's completely separate. It's not linked like centrifugal density gradient sedimentation or any of those other more bulk property measurements. So the basic principle said any means of sorting objects, biological or non-biological, according to an electronically extractable measurement.
Now, Becton Dickinson was the first commercial manufacturer.
Yes.
Did they have to receive a license?
Yes. At that time, I worked at Los Alamos. I worked for the Atomic Energy Commission. Well, let me correct that. I worked for the University of California, which was under contract to the Atomic Energy Commission. Under that contract with the Atomic Energy Commission, the commission had first rights to all patents coming out of work at the laboratory. The university had second rights to all patents coming out of the technology. The Atomic Energy Commission chose to take this patent, and at that point it becomes property of the U.S. government. And any American citizen could get a license to that patent at no cost. Non-U.S. citizens, there were some arrangements between different countries, but within the U.S., anybody could have access to the patent at no royalty.
Now, after the instrument was built, what sort of uses was it put to by the scientists?
When we were first at Los Alamos, we came up with this system, and then Marv took it on to develop the fluorescence sensing and lasers and so on. Even though we were in a biological group at Los Alamos, we had a hard time getting the biologists to really accept the idea of quantifying cell populations. The technologies, the sorting and the analysis, brings in the capability of analyzing whole populations of cells, hundreds of thousands, millions of cells, analyzing each of those cells, recording the measurements for each of those cells, and then describing the whole population in terms of percentages with different characteristics. And that idea is new to biologists. It was new to biologists at that time, so that it's difficult to get them to think in those terms. Normally they think in terms of taking a flask full of cells, grinding them up, and measuring an average value of some characteristic. So the idea of population studies was new. At Los Alamos, Marv had a heavy interest in DNA analysis. And so we, in our group and the other supporting groups at Los Alamos, began to look at DNA properties in malignant cells, non-malignant cells. I had a National Cancer Institute project to apply the technology to detection of cancer cells to replace the Pap test. The first biological group who could clearly see the uses of the method were the Herzenbergs in Stanford, who could see the application of fluorescence sensing and the use of antibodies in the characterization of immunological properties of cells and of surface properties of cells. So those were the first two heavy uses, DNA analysis and immunological staining.
Now, at this point, maybe we should break and then bring in Dr. Van Dilla, and he could take the story from there. now be speaking with Dr. Marvin Van Dilla, who, together with Dr. Fulwyler, was involved in the early research on cell sorting and cell analysis at Los Alamos. Dr. Van Dilla, how did you become interested in cell sorting and flow cytometry projects?
Well, as I remember it, it was accidental. I had been working at Los Alamos on gamma ray spectroscopy, a method of measuring small amounts of fallout radioactivity in people and in foodstuffs, which I think Mack mentioned earlier. And at the same time that this was going on, a pathologist in our group, in our health research group, by the name of Lushbaugh, was working on using the Coulter counter to enumerate red cells, and that had just been recently invented and made available to the medical community. And he wanted to do more than enumerate red cells. He wanted to measure their size and their size distribution to make population studies. And our group leader, Wright Langham, realized that this might be made to happen if he could get Lushbaugh and me together, because I had the instrumentation for gamma ray spectroscopy that could be adapted, in theory at least, to do the thing Lushbaugh wanted to do with the Coulter counter, convert it from an enumerator or counter of cells into a cell spectrometer that would measure the spectrum of cell sizes in the population. So Langham got the two of us together, and it seemed to me like, yeah, it was perfectly feasible, and it should work, and it shouldn't take long, and I had some spare equipment that I could loan him, and so I got together with Lushbaugh, and it took not too long to convert his Coulter counter into a Coulter volume analyzer, and or at least an analyzer of the signals coming out of the Coulter orifice, which we hoped were telling us cell volume. Well, it turned out that that assumption is a very tricky one, and it's easy enough to analyze the signals that come out of the sensor, but now what do those signals mean? And that's where it got sticky, because Lushbaugh was, as Mack described earlier, was using the Coulter sensor under conditions in which it gave spurious results, and so he was getting a signal distribution which had two bumps in it, which he interpreted as two different cell populations. When by that time Mack was in on the project, and the two of us thought that that was spurious, and the population really only had, there was only really one population, and he was getting incorrect results from the instrumentation, which is very easy to do. And so this got us, but this is a subtle thing to track down, and this got us both involved heavily in high-speed cell measurements, and using such high-speed cell measurements to trigger cell sorting, which was Mack's concept and Mack's invention. He described that earlier. So that's how it happened. Really, our group leader, Wright Langham, gets a lot of credit, should get a lot of credit in all of this, because he not only got these two diverse types of people together, but was very supportive and encouraging throughout the whole development. Although the way he encouraged it was to come down to the lab and look at what you were doing and get you to explain it in detail, and then shake his head and say, that'll never work. And that made you so mad that you were damn sure to get it working. But that was his style of management, and it was great, although at the time it seemed irksome.
You were very much interested in the phenomenon of fluorescence, and how were you using fluorescence in your work, and how did you see this instrument contributing to to your research?
Well, we originally started using cell volume or cell size or something about cells that was that we were extracting from the Coulter idea, from the orifice with the cell going through and the DC electric current going through at the same time. And the results of those experiments, what those experiments produced, what I'm trying to say is those experiments produced very interesting and illuminating results to a couple of the cell biologists around the lab. And a couple of them, not the whole gang, but a couple of them got very intrigued by the possibilities of high-speed measurements of cell size distribution. And we got to thinking about this and came to the conclusion that if measuring the distribution of cell size was important and useful in cell biology, that measuring other more important properties of cells, measuring their distribution over a population of cells ought to be even more important in biology. For example, the DNA content of cells is a very fundamental biological property of cells, and if we could measure the DNA content, which would have to be done in a different way than using the Coulter principle, that would be important. If we could measure other properties of cells, other cellular components like the cellular RNA or cellular surface molecules or cellular enzymes, that would be more important than simply the size of the cell. And so we got to thinking about all of this and came up with the idea that we should imitate the techniques that had been developed for fluorescence microscopy, in which you use fluorescent stains on cells on a microscope stage to light up a cell, to make a cell fluoresce under stimulation of blue or UV light, and use fluorescent stains that give you information about important cellular components. And one of the fluorescent staining techniques that have been developed for microscopy over the years was called the fluorescent Fulwyler technique, in which you could make the DNA of the cell light up and fluoresce and give a nice optical signal. And so we realized that optical sensing of cells was important to develop. So we set out to see whether we could adapt the fluorescence staining of cells that had been developed from microscopy to this flow system technique where we could flow them by and measure them thousands per second. And so we decided to move on from the Coulter principle, which had been the initiation of all of this, to optical measurements of cells, both the fluorescence of stained cells and the light that was scattered off the cell, which happens at the same time that you get the fluorescence signal. So we got interested in both the light scattered by cells and the fluorescent emission from stains that bind to important biochemical components inside the cell. The DNA was the one we focused on initially. So that was the thinking and the sequence.
Now that we've got the instrument opened up a little more, maybe you can describe in a little more detail the guts of the instrument, as you call it, and how it works. Okay,
well, the guts and the principle on which it works basically are all very simple. And this is the guts right here. This little flow chamber with the 4 optical ports that Boris Rotman described is where the real action takes place. And so what happens when, for example, you're measuring the DNA content of the cells of a cell population that you're interested in, And this cell population could be tumor cells growing in culture. They could be normal cells growing in culture. They could be sperm cells, which we spent a lot of time measuring. They could be cells important to immunological function, and you could be looking at cell surface properties, the properties of the surface of those cells, which is the thing that the Herzenberg group dived into very early and developed very beautifully. So you start off with a cell population which has been fluorescently stained to make visible an important cellular component. Let's talk about cellular DNA. So the cells that have been stained according to the DNA content come flowing from this little tube here up through the bottom of the flow chamber and into the flow chamber. And the flow chamber forces them to flow upward in a single file, one behind the other, at high speed. And that line of cells then is the object upon which the exciting light from this system here, which happens to be in this case a mercury vapor lamp, is focused. So as the cells move upward through on the axis, the central axis of this flow chamber, light, exciting light from this lamp comes in one of the ports and is focused on that line of cell flow. So every time a cell goes through the exciting light, the fluorescent dye in it is caused to fluoresce, emit a burst of fluorescent light. And that burst of fluorescent light comes out in all directions, including this direction, in which we have the sensor, the sensing system, which is a photomultiplier, which sees this fluorescent light, which is picked up by this objective lens and focused on the photomultiplier. The photomultiplier sees that light and converts it into an electrical pulse. So what happens is, as each cell goes through the light beam, the blue UV light, as each cell goes through, it makes a quick pulse of light that only lasts microseconds, because that's the residence time of the cell in the light beam. That quick pulse of light gets picked up here by the photomultiplier, converted into electrical pulse, which lasts microseconds, and then amplified in the preamplifier and the main amplifier, and then into the analyzing machinery, analyzing electronics, which measures the pulse amplitude. And the brighter the cell, the bigger the pulse. The duller the cell, the smaller the pulse. So cells with lots of DNA give you a big electrical signal, and cells with smaller amounts of DNA give a smaller electrical signal. And that distribution in pulse amplitude then reflects the distribution in DNA content of the cells. So if you have a population of cells, some of which are young and have a certain amount of DNA, and some of which are mature and have double that DNA, they're getting ready to divide, then you'll get a pulse height distribution that reflects that tells you that some of the cells are young and have the standard interphase amount of DNA, and another subpopulation has twice as much, and it's getting ready to divide. And then there's a population in between as representing those cells that are in the process of duplicating their DNA and getting up to twice the young cell value. So
Going back to the instrument, we have removed the stand that had the flask and the chamber that contained the cells, so that we could focus on the guts of the instrument, as you call it, the cell analyzer, and then at the cell sorting part of the instrument. And could you explain to us how these two parts come together and how they're used?
Yeah, the fluorescence or optical analysis device, which we call nowadays a flow cytometer, only analyzes the properties of the cell population that you put through it, which is extremely useful. But if in addition to analysis of the population, you want to separate out, physically separate out or sort out a subpopulation, then this optical sensing element has to be connected to the droplet formation mechanism of the original cell sorter here, which had a cultus sensor. So we have to get rid of the cultus sensor and substitute a fluorescence sensor for it. And that marriage was the thing that the Herzenbergs and their group saw would be very, very useful in immunological research, and did it at Stanford. And the resulting facts of fluorescence-activated cell sorter, which is a good name, was very successful, and that was the BD product, and that's the way most people do it these days. So with the optical sensor mounted along with the Doppler formation and deflection mechanism, you have the current version of the machine. And that version of the machine we used, and other people used, for a wide variety of biological and medical experiments. In addition to the measurement of DNA in proliferating cell populations like tumor cell populations, we spent a lot of time looking at the DNA content of other cell populations, like sperm cells, and we were quite interested in being able to detect in a sperm cell population the male and female sperm cells. That is to say, the sperm cells that contain an X chromosome and the sperm cells that contain the Y chromosome. And you can measure, it's only a small DNA difference, but you can measure that and actually sort sperm cells on the basis of whether they are destined to produce a male or destined to produce a female. And this was of great interest to agricultural people in animal husbandry, and still is. And that work is still going on at the Department of Agriculture right now. The other area that we developed after I left Los Alamos and went to Livermore, the Livermore National Lab, was the chromosome analysis and chromosome sorting area. And we did the first chromosome analysis and chromosome sorting at Livermore in the early 70s, about 74, I think it was. And what we did here is we took metaphase cells that had their chromosomes condensed into what are normally thought of as chromosomes, as seen in the microscope in mitosis. We took those cells with condensed chromosomes, mitotic chromosomes, busted them open so the mitotic chromosomes spilled out into the medium, and then ran those chromosomes as we ran cells one at a time through the axis of the machine, and excited them with fluorescence and picked up, excited them with UV and picked up the fluorescent light and used that to activate the cell sorter. And so in that way we discovered we could see individual chromosomes, which are much, much smaller and less fluorescent than whole cells. Not only see them, but see them with accuracy and sort particular chromosomes out of the population, which in the case of humans, there are 24 human chromosomal types. So you see these types when you run them through this kind of an apparatus, and you can pick out an individual human chromosome and sort 1 million or 10 million of them. And that enables one, that gives one enough DNA from the particular chromosomal type to clone. And a project was set up in '83 between Livermore and Los Alamos, both of which had this technology, to sort the individual human chromosomes in enough bulk for cloning, and to clone all the human chromosomes into DNA collections called DNA libraries, bad misnomer, and make them available to researchers who were studying genes on these particular chromosomes. And so that was an extension of flow cytometry and sorting into molecular biology and human genomics that has turned out to be very useful, and the project is in the final phases right now of the two labs.
Now, is it quite easy to separate the 24 different chromosomes, or are the differences quite small and the machines have become very sensitive?
Well, the early versions of the machines didn't have a chance with this, but as time went on, the resolution and stability and reproducibility of the machines improved, and not only did the machines improve, but even more importantly, the methods of handling cells and chromosomes, disaggregating, getting single-cell suspensions, keeping junk out, the staining techniques for cells and staining techniques for chromosomes, all slowly improved. And that's a very critical and important part of the whole business. Probably more important than the instrumentation development is the ability to produce a cell or chromosome population that's suitable for entrance into the apparatus. If you have a perfect apparatus and a lousy cell prep or a lousy chromosome prep, you get lousy results. It doesn't matter how wonderful the apparatus is. And that's hard to do. So with the improvements in the cell and chromosome prep techniques, and with the improvements in the apparatus, you now can see not only all the chromosomal types in people, but you can see homolog differences. That is to say, the chromosomes in your cells or my cells come half from our mother and half from our father. And sometimes the maternal and paternal chromosomes, take the number 1 chromosome as an example, which is the biggest of the human chromosomes. Sometimes the two chromosome types, namely the paternal and maternal number 1s, will be identical in DNA content and form one peak or one subgroup. But sometimes they're not quite identical. There might be a 1 or 2 or 3% difference, 4% even, in DNA content between the maternal number 1s and the paternal number 1s. But we can see those now, and so we can see in a chromosome distribution the chromosome that came from the mother and the chromosome that came from the father. And this turns out to be very useful for geneticists who are studying genetic characteristics and genetic diseases and genetic abnormalities. So the technique has been developed considerably since it was first worked up in the early 70s, and is very useful in not only in the Human Genomics Project, but in human genetics, medical genetics altogether these days. So that's an interesting combination of the technology, the physical optical technology of cell analysis and cell sorting by flow methods, and human genetics, medical genetics, and human genomics.
Thank you. Good summary. I don't want.
It's Friday afternoon, June 28, 1991. We're in the
a sixth-floor conference room of the biomedical building on the campus of Brown University, talking with left to right Doctors Mack Fulwyler, Marvin Van Dilla, and Boris Rotman. We will continue the discussions we had this morning about the development of cell sorters. This morning we concentrated more on the science and technology, and this afternoon we'd like to provide more of the context in which that work was developed. And I'd like to start with a discussion of your backgrounds, because I find it interesting that all three of you did important work in biology, and are still doing important work in biology and medicine, yet none of you were trained as biologists. Maybe starting with you, Mack, to give us a little précis of your scientific career.
Okay. I came out of Idaho State College in Pocatello, Idaho, with a bachelor's in physics, and the quality of the education that I received there was low. I went into the service through ROTC for 2 years and was exposed to a broader range of educated people and began to realize that my education was lacking. I applied to a number of places for jobs, and one of them was Los Alamos, which I had visited earlier while I'd been stationed briefly at Kirtland Air Force Base, and I had called Ernie Anderson and Marvin Van Dilla and asked if I could come up and just view their facilities, and they graciously arranged that and suggested I apply for a job when I got out of the service. So when I exited the service, I took the job at Los Alamos, went out there as a person trained only in physics, no biology, worked on whole body counting and fallout radioactivity in fallout detection and measurements, and began to go back to classes at Los Alamos through the University of New Mexico Extension System, heading for a PhD in nuclear physics. And partway down that trail, Marv and I started to work on the Colder system and on the ideas of analyzing cells in flow, and I came up with the cell sorter idea, which quickly excited my interest in the biological side of things. And I wanted to remain a physicist in the sense that I wanted to be technologically oriented, and yet I wanted to be able to bring that discipline into the field of biology and to quantify things, to measure things, and to try to bring to biology, cell biology, some of the quantitative aspects of the hard sciences, physics. So I went back to school. I had an opportunity to go to the University of Colorado Medical School, Department of Biophysics under Ted Puck, and get a PhD in biophysics. My thesis was the invention, development of the cell sorter, and application to a problem in cancer in mice.
This was on leave or something like that? From
Yeah, I was on an educational training program from Los Alamos Scientific Labs. They essentially paid for the first year of my schooling at University of Colorado. The other 6 months was on an NIH grant.
How about you, Marvin?
Well, let's see where to start. I guess start when I got interested in science. I had no interest in science, barely knew there was such a thing, through high school. But college, first year in college, there were a number of required courses to take, and one of them was called Science Survey, which was taught by an energetic and enthusiastic professor, and he was fascinating, and that turned on a strong interest in science in me that's never been extinguished, and that got me going in the direction of science, and it was physics, because he was the head of the physics department. I was going to be a physics teacher, but I never got to teaching. I got into research and got grabbed by it and never left. So after college and after World War II days, I decided to go to graduate school. Had a good opportunity to go to graduate school at MIT, and I got with the idea of getting a degree in physics. I did not want to. Those were the days when the AEC had just been formed and was flying high, and there was a tremendous amount of atomic weaponry and other kinds of weaponry jobs for physicists. I didn't want weaponry jobs. And so I looked around for a beneficial, socially beneficial application of physics, and the application of physical methods in biology and medicine turned me on, and there was a prophet in the physics department at MIT who was very strong in this field, and so I hooked into him and got my degree under him in what would now be called nuclear medicine, I think. And got out, and my first job was at the University of Utah Medical School in Salt Lake, and then Los Alamos, and then Livermore. In the Los Alamos experience, I got more and more interested in the biology and medicine part of this mixture that I was involved in, and I slowly found myself getting deeper and deeper, more deeply and more deeply interested in biology and medicine, and that continued at Livermore. And actually at Livermore, I took a year off, a year sabbatical, and studied biochemistry and molecular biology at UC Berkeley for a year. And so I'm some sort of a hybrid now between the two fields. I don't know what I am.
Just for the record, the college is City College of New York, right?
Right, City College of New York is where I went as an undergraduate, right?
Boris, you had an even more torturous road to science than that.
I wouldn't call it torturous because I enjoyed it all throughout. I received my education in Chile and went to engineering school in chemistry, got an equivalent almost of a PhD in the States in organic chemistry. And then I came to this country when I was 25 with the idea of staying 1 year and see the world and then go back. But I got a job in the laboratory of Saul Spiegelman in Illinois, who at the time was doing pioneering work with yeast and in genetics, what the infancy of genetics. He was famous for his plasma gene theory that later turned out to be wrong, but he managed to make the cover of the Time magazine on that theory at the time. So after
when you said
where
just speak
do
it
we
up
go
with
back?
Spiegelman. Just
Huh?
speak it up with your work with Spiegelman
all
and
right.
go on
All right.
from there.
So I came to the States as an organic chemist to work with Spiegelman in Illinois, and after 1 year of work, I was in love with microbiology. The laboratory of Saul Spiegelman was probably the most exciting place I have ever been. I always wished to have a group like he had. First of all, he was very loyal to his students. We went to every place he went. We had parties with the best scientists in the world, and so we got to know the best minds in biology through him. I got my PhD with him and then went to work with Lederberg in Wisconsin for 3 years, and after that I went back to Chile to teach in the medical school there, biochemistry. But I was the only microbiologist in the country, so I just found that I didn't have anyone to talk to, and decided to come back to the States. I went to Harvard for a postdoctoral and then had a job in Albany for a year and a half, and then Lederberg brought me to Stanford to work first at the Department of Genetics and afterwards at the newly formed Syntex Institute for Molecular Biology. Lederberg was the director of that institute. I stayed there for 6 years, and then I decided to get out of Syntex because at the time Syntex moved to the States as a pharmaceutical company, and in those days academia was considered much higher than industry, and besides I also like students, and so I accepted an offer I had from Brown and came here, and I have stayed here now for 25 years. With regard to my interest in biology, I started, as I said, with organic chemistry, then with yeast and bacteria, and then when I was at Stanford, discovered mammalian cells and got interested in the field of immunology because of our discovery of fluorochromasia. That was a new phenomenon, and I wanted to learn more about immunology. So I went for a year to the Karolinska Institute before Brown. Then I have been back there for 2 sabbaticals. From immunology, about 8 years ago, roughly, I went into cancer research, interestingly also because of fluorochromasia. So fluorochromasia has been the background of all my switching, and of course it was the reason Marvin called me in the first place. So
Well, let's talk about a little more about the context in which the cell sorter developed. And aside from the fact that all of you were trained originally not in biology, but they went into that work, I think people might find it strange that an instrument that's become such a major tool in the biomedical sciences was developed at a weapons laboratory. And maybe Mack, you can start it off, and then Marvin, you can add to why you think that happened there at that time.
I think it's an anomaly that it happened at Los Alamos. There was no stated need for a cell sorter. Nobody expressed the idea that that was necessary or important to biology. There was no process by which a proposal was written and submitted to a review board for consideration. It was a decision made by my boss, the boss of our group, Wright Langham, based on his opinion, substantiated by a few close advisors, that it was a worthwhile project to pursue. because it was considered a high-risk project with lots of opportunity for failure, before we did any work, I wrote it up, just a paper describing the idea, and passed it around to, I believe, about 8 different scientists, asking them to read it and give me their opinion as to whether or not it was a feasible idea, and they thought it could be made to work. The majority of that group of 8 said no, that they didn't think it would work. So in spite of that, which you could liken to a review process, and the negative outcome of that review process, in spite of that, Wright Langham had the courage and had enough authority to go ahead and launch the project. The project was never separately budgeted. I don't believe that it was separately budgeted. It was funded.
There wasn't any budget that I ever saw.
I don't think even in the following years that it was a separate budget item. It started off with a little bit of excess money out of Wright's budget, but we needed more money for fabricating some of these special parts, some of which contained platinum, were very expensive. And Wright was able to go to the laboratory director and get a hold of some of the discretionary funding that was available for weapons testing, weapons development. Also, we needed a good deal of special things made in the machine shops, and they were able to build us something for the self-sorter and charge it against a weapons account. So there was enough loose money at that time to allow this kind of a relatively wild, high-risk idea to be pursued. But if the 8 scientists whom I had selected to look at the idea had been an NIH review committee, we wouldn't have launched it.
Marvin, could you just talk a little bit more about the sort of the atmosphere at Los Alamos? Because it sounds that when you say weapons laboratory, that this is all sort of militaristic mentality, dominant, but from what you told us yesterday was quite different.
Yeah. Well, let's look at the Atomic Energy Commission and the atmosphere of the times in which it existed, namely the last half of the 40s and the 50s and on into the 60s. During this whole time, biomedical research was very important to the Atomic Energy Commission, and the reason it was that the people who set up the Manhattan Project during the war were very aware of the damage that radiation had caused to earlier scientists in the area, like Marie Curie dying of radiation injury, and the radium dial painters in the 30s in New Jersey dying of bone tumors in a few years from inadvertent radium ingestion. And so, and people being injured by, in the early days of X-ray machines in the 1910s and 1920s. So by the time the Manhattan Project came along in the early 40s, the hazards of radiation and that it could hurt you if you were careless had really been drilled in everybody's heads. And so when the Manhattan Project was set up in the 40s, 41 actually, a very important radiation protection biomedical component was set up to be sure that people who worked on it didn't get hurt. And so the biological hazards of radiation and the question, what does radiation do anyway when it falls upon living tissue and living cells, was very important from the very beginning. And so when the Atomic Energy Commission was set up in 45 as a civilian commission to take over all of this work, an important part of its function was this area of biomedical research, and the people who ran the AEC took a very broad view of the question of the biological effects of radiation, and they wanted to know all sorts of things about it, not just specific practical things. And one of the things they wanted to know is, what does radiation do to cells anyway? And part of the effect of radiation is on the DNA, so they wanted to know about cellular DNA, and they wanted to know about chromosomes, and they wanted to know about chromosome aberrations induced by radiation, and effect of radiation on the blood-forming tissues, which is also a very important part of the biological effect of radiation. So really, the interests of the old AEC covered a very broad field of biology, and they funded a great deal of biological research, not only in the national labs, but in the universities. In fact, in the 30s and 40s, rather in the late 40s and 50s, AEC funding in this country exceeded NIH funding of biological research. And NIH caught up and took over as it grew in the 40s. But for a while, the AEC was funding most of the biomedical research in the U.S. So the biomedical research programs at the national labs that the AEC ran, like Los Alamos and Brookhaven and Oregon and so forth, were all very important and well-funded from the beginning. So the atmosphere, and of course in the 40s and 50s and 60s, the AEC had lots of money. And so the atmosphere was very good for biomedical research and for trying out high-risk ideas, as Mack was describing. The cell sorting and the whole flow cytometry idea were kind of far out. But the atmosphere was very good for doing this. Also, there was another very good atmosphere at Los Alamos and at the other labs, and that was that a lot of the things that went on at these labs was physical and engineering type of activities. And so the idea of bringing the physical and engineering approaches into the biomedical research at the labs was something that was favored. And the physics and chemistry and engineering types around Los Alamos looked at the biomedical division very favorably and as a chance to contribute some of their stuff to biology and medicine, which had a very high reputation there. So we got lots of cooperation from physics and chemistry and electronics engineering types for whatever we wanted to try. So it was a really nice fertile field, although it superficially doesn't sound like it ought to be if you think of the labs as bomb factories. But they weren't and aren't now. That the weaponry work is a large part of what they do, but by no means 100%. And the attitude toward biomedicine was always very favorable and regarded with high importance.
I'd like to discuss for a few moments the dynamics of laboratory work at Los Alamos, and specifically referring to the work on the cell sorter. Maybe you could start out, Marvin, and give us an idea how you divvied up the tasks on the cell sorter, just to give us an idea of how that work went on and what interaction there was between the people who worked on this project.
Okay. Well, it started off by this project of lush balls, everybody called them lush, on using a very early model of a Coulter counter to first count the red cells, and then measure their size distribution. And I got roped into Lush's project because I was doing gamma ray spectroscopy on fallout radioactivity, and the kind of equipment I was using, multi-channel analyzers and photomultipliers and pulsite analyzers and pulse amplifiers and so forth, was the kind of stuff that theoretically could be hooked onto a Coulter sensor and measure the pulse size distribution out of the sensor. So Langham got Lush and I together, and I agreed to set Lush up with this kind of equipment so he could try out what he wanted to do with red cells. Red cells and the red cell forming organs being very important in radiation effects on human beings. And so I got going with Lush on this, and got him set up with some equipment to measure the pulsite distribution from his Coulter sensor, and he fiddled around with this and got some distribution curves for red cells that Langham didn't like the looks of, and showed them to us. By that time, Mack had come on board, and he got sucked into this, and we didn't like the looks of the results Lush was getting also. And so we got dragged back into it to try to figure out what the hell was going on. And this turned out to be subtler than setting him up with the original equipment in the first place, and took longer, and generated much interest in the two of us. And by that time, we brought Paul Mullaney into the lab from a teaching job he had in a little college in New York State, and he got involved in this general area. And soon we found ourselves not only heavily at work on the Coulter sensor and Coulter spectrometry. We found ourselves looking at other ways of sensing important properties of cells, and that got us into staining cells and using optical methods. And as these projects went along, things sort of sorted themselves out. Mack had the original idea for how to sort cells, and so it was natural for him to take the ball and run in that direction. Paul and I were very fascinated with the problem of measuring optical properties of cells, fluorescence after fluorescence staining, and light scatter, which automatically comes no matter what you do with a cell. If you shine light on it, you get scattered light. And scattered light is telling you something about the cell. And so somehow I got off along the fluorescent staining road, and Paul got off along the light scattering road. And so we had these three paths going on, these parallel paths going on simultaneously, with a lot of feedback back and forth, and everybody put ideas in back and forth, and it was a very, very neat environment. We didn't have to worry about funding. Somehow Langham liked it all and came up with the money, and it was all a little sort of paradise compared to the way you have to fight around for funding and grants and all that junk these days.
Well, let's skip ahead and there's an area that I want to discuss with all three of you, and that's the relationship between academia and the corporate world, and science that's being done in academia and the corporate world. And I guess you can also have a perspective of federal science as well. And I know that both Mack and Boris, at least, have been back and forth in both of those worlds. And I know that, Marvin, you made a conscious choice not to get into the corporate world. And maybe we just can go right in the road, starting with you, Mack, and talking about your experiences in both, actually three, because you were in the federal world too, and comparing and observing how science is done in all three, and addressing some of the concerns of, say, whether science is open in one and closed in the other, and to what extent that's true or not.
Okay, starting off with the working for the government. At Los Alamos, Marv will cover this in more detail, but it was a very unusual time, and it was a very fertile time for this kind of an idea. Marv will carry it from that point on in, so I don't spend any more time on that. After I came up with the cell sorter at Los Alamos, and Coulter Electronics saw that as a business opportunity. They wanted me to leave and go to work for them in Florida, which I did not want to do. I did take on a position as a consultant to Coulter Electronics, and I was a consultant consultant for several years. Later at Los Alamos, I developed a way to make very uniform particles, plastic particles, of whatever size one would select, from about half a micron to maybe 20 or 30 microns. So we could make particles of whatever size we wanted. We could make them in large quantities. Coulter Electronics also was very interested in that, because that would give particles which could be used to calibrate either the standard flow machines, Coulter counters, and you could also even mimic cells. You could make particles of a size that matched the size of different cell populations and mix them. In other words, you could make a pseudo cell population. So it was a useful device, a useful method to Coulter Electronics. Well, they came back and offered me a chance to set up a company in Los Alamos, since I did not want to go to Florida. And we did that. I left Los Alamos Scientific Lab and started up a company called Particle Technology, which was funded by Coulter Electronics.
What was this?
Let's see, this was 1971.
Oh, okay.
And so I started up Particle Technology, and it was a lot of fun, because I started off with a checkbook, and I was able to hire people, rent space, buy equipment, make all of my own mistakes. I wasn't stuck with anybody else's mistakes. And it's an unusual opportunity to build a group, and I enjoyed that a lot. I ran Particle Technology, which had two main objectives for Coulter. One was the production of these uniform microspheres. There were three. Another was the development of cell sorter technology for Coulter's main production facility, which is down in Hialeah. And a third was to investigate and expand the flow cytometry, again orienting it toward practical, commercial, ideally clinical uses. So I ran that until about 76, when Wallace Coulter made a decision to move Particle Technology to Hialeah, Florida. And I chose not to go with that move. So I left Particle Technology at that point and went to Germany, to the Max Planck Institute on a fellowship. Let me describe the research activities at Particle Technology under Coulter Electronics. Wallace Coulter, runs his company in a very different way from what most publicly owned companies are run, how they are run, in that his motivation, while on the one hand it is to make money, in my opinion, he also runs the company with another objective, which is intellectual satisfaction for himself. He likes to pursue ideas. He likes to try new things. And he would explore ideas and technologies much more adventurously than would a publicly funded company, where you had to justify to a board of directors or to a group of shareholders how you're spending their money. So it's unusual in that I was free to do a fairly wide-ranging variety of projects. That's an anomaly. I don't think that's the normal industrial situation. So I had a year in Germany with Tom Joven at the Max Planck Institute. I then came back to California and took a job as technical director of Becton Dickinson. Becton Dickinson Fax Systems, fax meaning fluorescence activated cell sorter systems. This was a small division of Becton Dickinson that was being run by Bernie Schuer. We started off with a small number of people, 5 people, and then grew from that up to what is now BDIS, which is kind of a child or grandchild of the fax systems. There was much, Becton Dickinson is a publicly owned commercial company with a board of directors with a standard corporate structure, with very clear profit motivation, with very short-term payoffs being preferred. I don't think they would say that they have any long-range research objectives, that they're all relatively short to bring products to market that have an immediate and visible need. So at Becton Dickinson, the range of things that could be considered was quite restricted. I was able to come up with a number of ideas on use of fluorescent dyes, and some of these were all patented, and a few advances in instrumentation. But there were a lot of ideas that came up at the fax group that I wasn't able to get corporate interest in and were unable to pursue. So that was one of the motivations for returning to academia. So I left Becton Dickinson in 1982, took a position as full professor in the Department of Laboratory Medicine at UCSF with the hope that I would be able to sell some of these new ideas to funding agencies and get funding to go ahead and try things like image analyzing cell sorter, which is one that I haven't succeeded in getting funding for. So I went back into academia at that point. I went into academia. It's my first experience in academia. I found it much more difficult to get funding than I had anticipated. I found that my skills and capabilities, which are really in innovating new technologies and new devices and new approaches, didn't fit very well with what NIH saw suitable for funding. From my perspective, NIH was most interested in funding projects that were directed against a specific disease state, for example, or a specific area of basic research. And they were not particularly interested in funding a development of a device, development of a technology. So I found it, and this was true up until 2 years ago, found it very difficult to get NIH funding for my research. I've come back into the industrial world now, having retired out of the University of California. And there's several motivations there. One is that you spend an awful lot of your time writing grant applications and trying to sell your ideas into the academic world. That would not be so true for someone who had a 10 or 20 year string of grants, and so the next grant is an extension, continuation of previously funded research. My situation was to come in from industry and to try to get started from ground zero, and I found that very difficult. One of my motivations then was to get out of the grant writing cycle, which is frustrating. And I found I was also looking for the excitement that an industrial startup offers, because things move so much faster. You can change directions in 24 hours instead of 24 months, and the stresses are much higher. But the pace is exciting. The pace is exciting. Again, I'm constrained to developing things which fit the corporate objective, but the particular field that I'm in now, which is this extraction of pancreatic islets, encapsulation in a semi-permeable membrane, transplantation as a cure for diabetes, has got so many problem areas that need a new approach that I see lots of fun, exciting opportunities to do it. Places where I can do my thing and make a useful contribution to this still primitive field of tissue extraction and encapsulation.
Well, Marvin, I leave you, and you decided that you didn't want any of that. And tell me more about why you just wanted to stay in the academia and in the federal laboratory.
Yeah. As I look back on it, I realize now that landing in the three jobs, the Salt Lake City job, the Los Alamos job, and the Livermore job, were all in the same kind of a laboratory, namely a national lab setup or something rather close to a national lab setup. And I realized, now that I look back on it, that it must have fit me very well. This is national labs are sort of like academia in some ways, but without the teaching and without the close association with the students. But researchy wise, very similar. And I realized that must have fit me very well, and because I was happy doing what I was doing, and there seemed to be, and I was happy living where I was living. There was no good reason to make a change, and so I didn't. So I guess that's all there is to it. I was happy in that environment, and it was working well for me, and I think I was into good projects with in the company of damn good people, and the funding situation wasn't as wild and frustrating as it is in academia, and so why fix it if it ain't broke, I guess. Must have been my general point of view, but I never really thought about it too much at the time, I guess.
Boris, you went back and forth. Are your reasons similar to those with Max, or did you have different motivations?
Well, Mack's career parallels a lot mine, except that I went in and out for different reasons. I, in essence, fit more Marvin's pattern that I didn't like industry, really. The reason I went to Syntex was first that Lederberg was running it, and one of the first things that we were told, by the way, Carl Gerasi was a co-director of the institute, and he is professor at Stanford also. And we were told, under no circumstance we want you to do applied research. And that was very nice, because we had all the money, no teaching, close to Stanford. We had faculty appointments at Stanford, so we had the best of both worlds. It was only when the industry of Syntex came out, and one of my expressions was, I can smell industry on the corridors now. When the money-oriented people came in, I had the problem. And at the time, they offered me to stay in the Syntex, but I was told, if you want to go places here, you have to become an executive. And they would give me stock options, and for 5 years when I came to Brown, I would look at myself in the mirror every morning and say, Boris, you are stupid. Look at all the stock you would have had. Syntex stock went like this, about 80-fold in something like 5 years. 80-fold. So I was stupid so I had a hard time convincing myself, Boris, you are clever, really. You didn't want this dirty money. You are happy now correcting exams and doing things. At the time, I was rolling in money at Brown. I had 3 grants. I was working on 3 different projects, getting funded in all of them. It was very exciting. And the similarities with Mack came later on when the grants start drying out. I have no problems getting money in the field I have been all my career, which is transporting bacteria. But if I wanted something new, I had to sneak it into my research, and got to a point where you couldn't do it simply. You couldn't have a new idea and work it into your grant without risking losing the grant. And I was maybe silly enough to do it, although that turned out to be really what I consider my most important work, which is the present work on cancer chemosensitivity, which has the potential of helping millions of people. Basic scientists have the dogma. We always believe our science eventually will help people. But when I was about 50, 55, I start looking at my papers that say, how is working E. coli on beta-galactosidase is going to help anyone when no one even knows except myself and my sister that I have written those papers. And so I was very glad to jump into this new research, and like Mack, I found very exciting going into industry. Brown got money from a venture company, and we set up a large laboratory to make this test available to oncologists. But somehow the glamour of going around in plush hotels and eating good dinners, traveling in first class and all that starts wearing off. And again, one day I look at myself in the mirror and I say, I don't want this. I didn't really like the business aspects of the company. One of my principles has been I don't like to be owned by anyone, and that's why I left industry. I hated the hierarchy that I have to answer to my boss, although my boss was the president of the company, but still, I don't like to answer to anyone. And that's how scientists, basic scientists, are used to. Peer review is another story. But you want someone to call and say, Boris, what have you been doing in the last 6 months? So that was my major reason. So what I finally did, I got out of industry by giving all my stocks of the company to Brown University, and now I am associated only as a consultant, and I am very happy not to deal on the daily problems of the industry, although as Mack said, they were exciting at the time. But in the long run, I really like my laboratory.
What was the name of the company?
Is the name of the company.